So today, my talk is about combining XAS and DFT methods to connect spectral features to the electronic structure of bioinorganic. And what I've added is electrocatalytic systems. So I wanted to start at bioinorganic system and then end with an electrocatalysis system. I've been at SSRL now for about 15 years, and I've been doing X-ray spectroscopy for a little over 20. And combining XAS with DFT methods is very close to my heart. And I have structured this presentation a little bit in terms of, giving you those examples that I've chosen, but also trying to rally the troops into doing more of this type of work. All right. So because the audience is very diverse, I wanted to give a small primer on metals in biology. The metals are extremely diverse in biology. They're ubiquitous from plants to animals to microbes. And this slide kind of shows that even though they're ubiquitous, they're present in very trace amounts. This example is of composition is for the human body. We are 99% made up of 6 elements: oxygen, carbon, hydrogen, nitrogen, calcium, and phosphorus. And the rest, almost 1%, is made up of 5 elements: potassium, sulfur, sodium, chloride, and magnesium. And a very tiny fraction is other essential elements, and they're essential because without them, we would not be around. Because of the nature of the world that we live in, we also have toxins and other metals in the form of environmental agents. Now, these essential elements are often these transition metals that are highlighted: manganese, iron, cobalt, nickel, copper, zinc, molybdenum, tungsten. These transition metals come in the form typically of metalloproteins and cofactors. And the entire world of bioinorganic chemistry tries to understand their structure function. So metals do play a very diverse role in biology. Of all the proteins, 30% of them actually have a functional metal center. And these functional metal centers do perform a wide range of chemical reactions. We know vitamin B12 is, a cofactor, which is very important for life. Then there is iron, and in these examples is iron and ferritin, which is used for biomineralization. We all know about nitrogenase, which is important for biocatalysis, etc. And you can see some of the other examples I mentioned here. Essentially, understanding the functional roles of metal centers is really important for us to further research, not only in biology, but also in medicine, bioenergy, environment, and catalysis. So what are the systems that fall into this wide box of bioinorganic, biomimetic, bioinspired systems? I'm choosing a few examples here. You start with metalloenzymes, and then the understanding that you get about the electronic structure and function of these enzymes can be translated to other more practical areas, such as biohybrid functional materials for catalysis. You can use those principles in green chemistry or other bio-inspired chemical catalysis. And a lot of research groups around the world do just that, take some of this understanding that we get from metalloenzymes and then apply them to these diverse areas of research. And I wanted to highlight here that because these metalloenzymes have these metal centers in them, which are interacting with proteins around them, they're typically coordination chemistry compounds or often organometallic compounds. So this talk is going to revolve around these type of systems that are relevant to bioinorganic chemistry. Now, I wanted to go back in time and give you this publication that came out in 1975. It was the first application of XAPs on metalloenzymes. It was in 1975 by Schulman et al. What they did was they determined the iron-sulfur distance in rubredoxin by X-ray absorption spectroscopy. This was done at SSRP at that time, which eventually became SSRL. And this rubidoxine active site is an iron center. So this is this iron center, which is bound to 4 sulfur ligands. And in those days, they showed that from the Exas data, they compared the rubidoxine data to a model complex, which kind of looks like this. And they made a very important discovery. They showed that the Exas data disagreed with the crystal structure and actually gave a better description of what was going on in the active site of this enzyme. And that really brought the importance of XAPs to the forefront, because at that time there was a debate that you have XRD, why do you necessarily need to apply XAPs? Here was the study which said we can actually correct some of the data that you get out of XRD and get more. And so that was, after this first study, there was this explosion in bioinorganic chemistry applications. Lots of important enzyme systems have been studied, characterized, and there are professors all around the world, or researchers all around the world focused in that area now. So today, the biological X-ray spectroscopy really involves a more in-depth, holistic look at the system. We look at the near edge zones, which is shown here, and that gives us covalency, bond strength, and type of ligands. We look at the structure, which of course, as you know, gives us information on the ligands, how many, what type, how far, and then a big push towards combining theory, where you add theory to get detailed electronic information about the site, and then together with all of these, you get structure function correlation. And what is more important that I won't go into necessarily in this talk is that it's very important to add other spectroscopies to this type of analysis, where you bring in EPR, Endor, Mossbauer, or other UV-Vis or other such methods to really understand the electronic structure from various perspectives. So that's typically the status quo now. Bioinorganic system. In fact, if you see a paper which only talks about XAPs and does not talk about the near edges, it will be certainly in the minority. Okay, so moving forward, again, for those of you who are not familiar with biological systems, it has a complex ligand space. Here on the top are some amino acid ligands, showing that just the amino acids can coordinate to the metal center through sulfur, oxygen, nitrogen. And on top of it, there are these other very cool ligand systems. These are these ring compounds. The porphyrins or the heme systems are the most ubiquitous, but we also have this chorin and corphin rings, which contain these cool molecules such as cobalamin and this nickel containing ring. They have a very unique X-ray absorption and XAPs spectra. And in addition, there are some other cofactors and other ligands, which really make the biological ligand space really complex. In this active site of hydrogenase, you can see that there's actually inorganic CO and cyanide bound, forming an organometallic compound within the protein. And in this example, I think, is a heterometallic iron molybdenum compound that's present in nitrogenase. So a variety of ligand systems, and as you can see, there's really hardly any order to these ligands. Now, the way I like to look at biological systems is I have my own categorization system. There are the good systems, which have typically single metal centers. Often they have heavy ligand coordination, have low multiple scattering, and you can get high K data. And in that, in my system, fall the molybdenum and tungsten containing enzymes, some zinc enzymes that contain cysteine ligands, and iron sulfur enzymes. You typically have a single first shell feature, like you see here. I mean, this probably is not the best example, but certainly the molybdenum tungsten enzyme and the iron sulfur clusters fall in that category. You can get very high K data and typically easy to analyze XAS. And then you have the bad systems, which have complex multiple scattering, multiple absorbing centers, flexible and weak ligands. And heme enzymes fall in that category because they have this complex multiple scattering, and you really need to fit the entire data to get meaningful information out of it. Then there are these trinuclear and tetranuclear copper enzymes, which essentially give you an average spectrum, and then you have to tease out the information that you want about one particular copper there. And then I've also thrown in cytochrome C oxidase in this because there are loading issues. In proteins, you can have partially loaded copper sites, and that would really mess up with your Exas data, and you have to be careful about the analysis that you're doing. And finally, there are these downright ugly systems, a complex multiple scattering, multiple absorbing sensors, flexible weak ligands, multiple oxidation states, very low concentration, and they're very beam sensitive. You put it in the beam and they go away. And I absolutely admire the people who work in this area, such as photosystem 2, hydrogenase, particulate MMO. But having said all of this, I think the typical bio-XAS experiment has the entire cast. It has some good, some bad, and some ugly about them. And I note these several different criteria at the bottom, which can either be good or bad for biological systems. And so they're typically a mixed bag. And in that sense, I don't think it's any different from other areas. Like environmental applications of XAPs also have typically a mixed bag like this. The other thing about biological XAPs, which may not be very obvious, is that because everything is in solution and everything is very dilute, you have a lot of flexibility in the XAPs signal. So here in the top, I'm showing you an iron heme enzyme structure, active site structure, where the iron is in the ferrous state. And to the right, I'm showing you the spectra of various ferrous heme proteins, yeah? This is just the Fourier transforms, and you can see this wide variability in the data. And that's because this heme ring is flexible. So you're thinking about this metal center in solution, which is, floating around, and you get this a very diverse or different looking spectra from sample to sample, which are in principle very similar to each other. So in biological systems, again, the XAPs can be weak or noisy because concentrations are low, and interpretation is often limited to first shell. And in some cases further out, one of the strengths of biological XAPs is really nailing down metal-metal interactions. So if you're looking at multi-metallic systems, you can really identify the metal-metal distance in there. Now, having said all of the negatives about doing biological XAPs, one big advantage about biological or bioinorganic systems is that the enzyme is in solution and the substrate is in solution, which makes it a homogeneous system. And we are looking at molecular level catalysis. And because it's a homogeneous molecular catalysis, you can try to get more quantitative information out of it from near edge interpretation. So we can look at the rising edge data and very cleanly assign them to oxidation state changes, ligand field splitting, etc. And so I just wanted to go through that a little bit for this audience and go over the information content in an XAS spectrum. So typically an XAS spectrum would look something like this. You would have a pre-edge region, you'd have a rising edge region here, and then you'd have the white line and further out. And if I map that to the schematic of a molecular orbital schematic of a cartoon molecule, you would see that the pre-edge region, which is right here, is transitions from the metal 1s to the metal 3d level, which has overlap from the ligand 2p3p level or the metal 4p level. Then we are talking about the rising edge, which has dominantly contributions from the 4p level. And then further out is the continuum level. And we know that this pre-edge level is, electric quadrupole allowed, dipole forbidden, and that's why it's so weak. And as you start going up here, you start getting dipole allowed character, and the features become intense. This is just the opposite for L edges, where the pre-edge is dipole allowed and the post-edge is a delta L of -1. And so the intensity is flipped when you're looking at L edges. Okay. So the other reason for looking at the Zane's data more and more instead of XAS is because you can collect the data really fast. Biological systems photo-reduce, and sometimes you just can't get good XAS data before the sample is gone. So it makes sense to try to get as much as you can out of the Zane's region. And the pre-edge region has been used very successfully to get ligand field strength and geometry, spin state, centrosymmetry, and covalency information. And the rising edge can be used to estimate geometric structure, as you know, from PEP or MX-SAN-based analysis, metal ligand overlap via shakedown transitions, ligand arrangement, and what is the most used use of the Zane spectrum is charge on the metal center. Most people will look at the edge shift, and say, okay, this is an oxidation state of X or Y. That's the most popular. Now, the XAS edges are qualitatively analyzed by most people. We use the edges as fingerprints of the electronic structure. And we say, okay, feature shifts up in energy or intensity is attributed to some literature precedent, right? That's what we do. We also do other methods which are a little bit difficult to apply to biological systems, such as, compare to known or related model complexes. Now, typically, you won't find any known complexes for these very unique biological centers. And using LCA or PCA analysis is also quite difficult. Because bioinorganic systems often are molecular, as I was mentioning earlier, we can approximate them to a molecule, in which case we can start doing molecular orbital-based approach and get all of these various detailed information about the electronic structure and bonding some of which I mentioned here. I should say that we, cover some of this in our summer schools that we organize every other year. We'll do one in 2022, and I'm taking this opportunity, Jerry, to advertise that we'll do this again in 22. All right, and you're welcome to visit the YouTube channel. We have lots of material there, sample preparation, and some other data analysis, both for XAS and imaging. Okay, so I've got one break here. Jerry, if people have questions.
Sure. First, Satish, if you're still here, do you have a question? He said he had to leave, but
yeah, Riti, that's very nice introduction. So, in one of the earlier slides, so you mentioned, good, bad, ugly, right? So, I'm curious how you would take the ugly part of this spectroscopy, like where you have sample changes and all that beam-induced changes, to interpret the good stuff.
So let me go back. So it's very, very system dependent, right? If you had photosystem, you would. So people who work on photosystem, they are lucky. The good part there is that they can generate a lot of protein, and so they have figured out methods where it's a very fast photo reducing samples that really do defocus the beam, and they collect these single scans that look super noisy, but they can collect hundreds of scans to average them in. And they then replenish their samples very, very fast. And so that's one way of dealing with, the beam sensitivity. Beam sensitivity is the worst, because if you don't have enough samples, then you're stuck. You just put in the beam and you measure it one time. But ideally, you have high concentration. The other way that people try to do this is try to look at HERFT and HERFT XAPs of these multiple metal centers, and that sometimes gives them a clue. If you sit on one region of the K beta spectrum and then collect the XAPs data, that might give you a clue on these multiple sites. But typically, again, it's very system dependent. You need lots of sample, you need lots of measurements, and you would also infer by measuring various different oxidation states, various different redox centers. So you could measure things in the oxidized form or reduced form, or super reduced form, and from there infer things, I guess. I don't think I'm giving you a very good answer, and I try to stay away from these systems is all I have to say.
No, but thank you. I mean, that's an important thing that, we in geosciences and environmental community have to think about all the time.
Yeah, and absolutely. And for geosciences, you also have these multiple peaks. You have other metals that are right next to it. So I appreciate the issues you see too.
All right, very good. You should continue, please.
Okay, all right. So, given, in the interest of time, I'm going to go fast and just say that I'm going to give you two examples. One on acetyl-CoA synthase, which I'm working on right now, and the other on a cobalt-based hydrogen evolution catalyst. And essentially, these two examples that I've chosen are connected to each other by being distillable into molecular systems and being organometallic compounds. So let's start off with the metalloenzyme catalysis example. I'm looking at a process called the Wood-Youngdahl process, which involves several metalloenzymes that convert carbon dioxide into chemicals and bacterial biomass, essentially. So you start with CO2, you form something called acetyl-CoA, and which is then upregulated or used by plants. And the enzymes that are involved in this process are very unique. There are 3 metalloenzymes. One is here, which is the CFESP protein. It's a methyltransferase, and I'll come to that. Then there's a CODH enzyme in the middle, which converts CO2 to CO. And then there's this other enzyme which takes this CO and converts that to acetyl-CoA. And that's why it's called acetyl-CoA synthase. Essentially, what happens at this active site of acetyl-CoA synthase is that one CO molecule comes in, a methyl group is transferred from the CFESP protein here, and they are fused to form acetyl-CoA. So the active site of acetyl-CoA synthase looks like this. This is a truncated schematic. It has a very nice looking iron sulfur cluster. For those who are not familiar, it's a beautiful cubane that's present in biological systems. Again, ubiquitous in biology, is mostly is there for electron transfer, and has these alternating iron and sulfurs in the cubane. So this cubane is linked or connected to 2 nickels. It's covalently bound through one of its cysteines to 1 nickel, which is the nickel P, because it's proximal to the iron sulfur cluster. And then there's another nickel, which is nickel D distal, which is further away. And I'm going to approximate that to this diagram here, where the iron sulfur cluster is here, the 2 nickels are shown this way. And a lot of study has been done on this already, which I will keep referring to as I explain this. But basically what's happening is this is in its resting form in these oxidation states. You throw in an electron, it gets reduced. Nobody has really characterized this reduced, 1 electron reduced form because that species is not trackable. You put an electron, nothing happens, still a substrate binds. And when the substrate binds, the next process occurs. So you take the electron and then you get the methyl transferase to give a methyl species here, and that will go and bind to the active site. We know that it's not binding to the nickel D site, which is there mostly for structural reasons, but we know that it probably binds somewhere here, nickel P, or the iron sulfur cluster somewhere, maybe a bridge. This is when I came in to do this research. Oops, sorry. And then CO will come in and CO will bind, CO will be fused with the methyl group to form this acetyl group. sorry, I just keep doing that. I'm sorry. Okay, yeah, it's fused, and then acetyl-CoA is released and the starting form, the reduced form, is regenerated. And the cool thing about ACS is CO or methyl can bind first, so we could have bound CO, brought in methyl here, and then CC fusion would happen. And when I started working with Steve Ragsdale on this, we didn't know how CO bound or how methyl bound, and those are the two species that we set out to characterize. So because this site has both an iron and a nickel centers, we can look at them from both iron XAPs and nickel XAPs. And I'm comparing the Fourier transforms here to the left, and you can see there's really no change in the iron XAPs when before and after CO binds, but there's a big change in the nickel XAPs before and after, both in the first shell and in the second shell. And that clearly tells us that CO binds to nickel, the nickel P center, and the distance was around 1.75 angstroms. But that was the extent of information we could get from the XAPs data. What has bound and what is its distance, right? We then turned to DFT methods, which actually showed, oh, I should go back here and say that the XAPs also showed that one of the nickel sulfur distances had elongated, but we didn't know which of the nickel sulfurs, right? So we went and looked at XAP, we compared the XAPs to DFT, and DFT revealed a lot more about it, which said that the nickel center, which was square planar, is now a tetrahedral with the nickel carbon bond at 1.75 angstroms, which matches the XAPs data really well, but also told us that the sulfur that got elongated was this sulfur that bridged to the iron sulfur cluster. We also did TD-DFT simulations of the pre-edge region, and we got a really good match for before CO and after CO binding, essentially making the DFT calculation really calibrated, both from a structural perspective and electronic perspective. And then we could look at charge distribution from the DFT calculations and show that the CO indeed binds at nickel P, and the nickel P remains in the nickel 1 oxidation state and becomes tetrahedral. Now, the question here was that the iron sulfur cluster really did not change, right? And when we look at both from the XAPs region and from the Zanes region, there really is no change in the iron data. And when we look from a DFT perspective and start looking at charge distribution, there really wasn't a big change in the charge on the iron sulfur cluster. So the question here was, there was no obvious role for this iron sulfur cluster in CO binding. And so we thought, well, it probably plays a role in methyl binding, right? And here I'm going to have to go in a little bit detail about the mechanism. So I apologize for this complex mechanism at the bottom, but let me walk you through it. So you start from the resting active side, you add a reductant, and then you add a methyl transfer agent where the methyl goes and binds at the active site somewhere, right? Now, nickel 2+ is D8. This nickel 2+ is also D8. So both of these are spin of 0 species. Iron sulfur cluster we know is S = 0. When you add an electron, you should get a half integer spin species, which should be either EPR active, or you should see the signal at least in your Endor or Mossbauer data. But it turns out Endor and Mossbauer spectroscopy tell us that the methylated intermediate that we are generating is actually diamagnetic. There is no change in the iron and there's no change in the nickel from a spin perspective. And so I'm kind of letting the cat out of the bag in the beginning a little bit, but I thought it was important to establish it in the beginning, otherwise it may become confusing. We are arriving at a mechanism where, again, we start from a resting form, which is, iron 2+, nickel 2+. We do a 1 electron reduction, which this electron reduces it to this form. we add a methyl transfer agent, and we're supposed to form this species, which should be half integer spin, but because we have excess reductant, it gets reduced to a spin 0 species. So our interpretation based on all of these data is that we are dealing with a spin of 0 species, and we want to characterize that now. So having said that, let me point out to what's really cool about this system. We start with a system which has both iron and nickel in it, and we are adding a methyl transferase which has a cobalt center in it. So we could, on the same species, same sample, we could measure all 3 edges, yeah? And so we started the methyl cobinamide, which is essentially a very cool molecule in itself, is the methyl transferase. We looked at the data for this and this, which is essentially reacted with to form the methylated species. We see a big difference in the pre-edge region, and this qualitatively matches what you expect when you measured methylcobalamin and the cobalamin in oxidation state of 1. There are some qualitative differences in both of these. We understand that really well. It's because of a transaxial ligand. And if you're interested, we can talk about it offline. But essentially, it's clear that all the methyl has been transferred to the ACS active site. Now, if you look at the nickel K-edge XAS data, we see a very beautiful pre-edge shift, changes in the rising edge region, and big change in the Exas region, which again clearly tells us the methyl binds at the nickel P site. And when we match it to the DFT structure, in this case, we start with the square planar molecule, which stays square planar. And the distance, it matches the Exas really well. We do TD-DFT calculation, again, very good match of experiment and theory. And essentially, we say that the methyl group binds at the nickel P site, it's a long nickel methyl bond, and there's only a small change in charge at nickel P. Now, if we again look at the perspective from the iron sulfur cluster, there's some change in the iron sulfur cluster, both in the edge and small one in the pre-edge, but it turns out that that is mostly structural, and a very small structural change that too. So before methylation, these are the bond distances that fit the 2 peaks, and after methylation, this is what it looks like. So probably a small change in the structure, but not a big change. And again, TD-DFT matches the data really well. And I should mention here that we're looking at a very large molecular complex. It's almost 300 atom complex for this calculation. So then it turns out that why does nature choose an expensive and elaborate iron sulfur cluster if it's not doing anything in CO binding and methyl binding, right? Like, what's its role? And so here is where we really turned to DFT to try and look at this intermediate, which we don't trap, but we feel gives us a clue to the role of the iron sulfur cluster, right? So what we are saying is that we do this reaction, it immediately goes to this intermediate, the reduced intermediate. But when you calculate this oxidized intermediate, we see some reasonable changes. So we went from the resting to methyl bound, and we take the methyl bound and oxidized it, we suddenly see a lot of changes in the structure and the electron density. And I'm going to skip over a lot of things and essentially just say that when you look at the ground state wave function of that oxidized species, it turns out that most of the oxidation is occurring in the iron 4 sulfur 4 site. So the whole sits at 60% of the whole is on the iron 4 sulfur 4, some of it is on the nickels. So essentially what this tells us that this transient species 4, which we haven't tracked, but we can look at it with theory, is a delocalized species, and it's really justifies the use of the iron sulfur cluster, which is there to delocalize the excess spin or hole on the nickel P. Okay, so this is done. I'll take a question. Do I only have 5 more minutes, Jerry?
No. We'll finish up at 10 o'clock.
Oh, okay, great.
Yeah,
All right,
the
sorry.
idea
So,
is there'll be the questions will flesh out the 40 to 45 minutes you prepared.
okay, all right, that's great. So yeah, so this is it. I end the story by saying we now know, thanks to the help of theory, what is the iron sulfur cluster doing there. And
Okay. So
yeah,
we got a
any
question
questions?
too. All right. Yang Hai, you had a question?
Yeah, so I actually have multiple questions, really. This is really a
Hi,
fascinating
Yang.
work. Yeah. So the first question is, I assume there's no existing model complex for this, like nobody can synthesize such a complete molecule or whatever model. So all your data or analysis were based on protein structures, right?
There are some models, very few, that kind of mimic the site, but not exactly. And there are some other, so some of them structurally mimic really well, but then don't do the function. And some others, they don't structurally mimic at all, but do the function.
It's always the case.
Yeah, exactly.
Okay, and you also showed that your DFT model is pretty big. It's more than 100 atoms. So any tricks to deal with such a large system in DFT? Did you do QMMM or just the traditional?
So let's just see. I think we were just below the limit where we had I had up to 300 atoms. And what we did to, I don't know if I can find that in here, right?
Yes.
So yeah, in this model, what we did was we, of course, fixed the C alpha.
Alpha C, yeah.
And so there are about 20 or 30 of them, I don't exactly remember, between 20 and 30 C alphas, which we fixed. And it was a little bit tricky because some of them did not like being fixed, right? And so we had to step up. We started off all being fixed, and then we could not, energy minimize, and then we had to float some of them, and slowly we iteratively and rather painfully came to the realization that there was some of them that we could fix, some of them absolutely needed to be floated. And once we found that, the calculations were faster. But you absolutely need a dedicated theory cluster to do this. I've built one at SSRL, as you know, through NIH funding. It's a 1,000 CP. It's not very big, but it's dedicated for this work. So you can't do it on a small 8 on a cluster.
For these multiple metal centers, you have to force their spins at some point.
Yeah, we started off doing that for the oxidized. So the iron were all initially forced, and then after that, once you find that electronic structure, you can just use that as your starting, and then that works. But that's a very good question, because you have 2 nickel centers, you have this iron sulfur cluster. Yeah. They can be coupled too, their spins can be coupled, so it becomes a very complex problem. And right now, our collaborator Simone Rauguli at PNNL, he's mapping out the entire spin combination space in this large molecule. I mean, we found one and we ran with it, right? But they're doing the big analysis on that.
Okay, last question, a general one. The CO2 reduction is now getting very popular, the negative emission thing. And most people are doing it with electrode or solid state catalysts. So can you just comment on your molecular system versus those solid state systems?
So actually, it's really cool. My next example is on an electrocatalytic system. It's not on CO2 reduction, although we also worked on CO2 reduction catalysts. I think one thing that I'm not yet involved with, but Steve, what he's working on is immobilizing the ACS with a material compound and some kind of MOF where you put the ACS on it and then try to use this biohybrid material for CO2 reduction. It's still not known why nature chooses this very complex active site to do this. right? I mean, I guess that's at the root of your question, is that so many small, simple molecules can do CO2 reduction at the right temperature and pressure. Why are you using this complex site? And that's probably part of why we are studying this. The one clue to why it has to be this complicated is that CO2 is a gas, and you actually forms carbon monoxide, right? You have to reduce it to carbon monoxide, and then you find methyl. And you don't want this carbon monoxide to be released out of the protein. So the entire process, is you see how CODH comes and binds ACS to transfer that CO. It's very tightly regulated. And there's both structural protein regulation and also geometry regulation. It's fascinating. And so maybe that because it has to be so tightly regulated, the structure is also gotten complex.
So you think the iron sulfur cluster plays a similar role than in nitrogenous, like in the P cluster?
I don't want to comment on that.
Okay.
I could say yes, and I would be very, very wrong.
Okay, thank you.
Yulia, you have a question or a comment?
Yeah, so this is Yulia Pushkar from Perduriti. Very impressive stuff. So I see in your mechanism potential oscillation between nickel plus and nickel 3+. I'm wondering, those in situ intermediates are really hard to trap, and also you have two nickels. How much of the age shift you would have between, limiting cases?
What is limiting cases, sorry?
Something that you approximate as nickel plus and something you might approximate as nickel 3+.
Right, so, that's go back to my nickel spectrum. Oops, I think I need to be smarter about navigating animation. So one more, and here, right? So we know our entire analysis is based actually on knowing through a large body of evidence that the nickel D site, this nickel D, does not change. That stays nickel 2, and that stays square planar. And that's a great point. What you mentioned was we are only seeing change on one nickel site when the other stays the same. So this is an average spectra. This is only 50% change, and yet you see a dramatic change both in the XAS region and in the Zanes region. And that makes us even more confident that we are trapping the methyl-bound intermediate. And if we did not know that this nickel D was indeed going to stay nickel 2, I think what you are saying would have become a big challenge for us, because we wouldn't know which one of them is getting reduced, to what extent, is one of them nickel 2, one of them nickel 1, etc.
And this shift, so how do you interpret before ME and after ME? So because I don't know details of your mechanism, so what is your interpretation of that spectral change? Because you have a shift and you also have a pre-age change. So if you
right, so let's look at this. So this pre-age change, which is shift up in energy and increase in intensity, is because you now have a strong nickel methyl bond at 2 angstroms, which replaces a water molecule, which is a weak ligand. So your ligand field is changing, and so your ligand field strength increases, and that's why it shifts up in energy. And because it is a strong overlap, you also get increase in intensity, because now you have more 4p mixing into that. So that's for the pre-edge. For the rising edge, again, this is very qualitative. This is what we have observed, as I said, I've been doing this for quite some time. Every time you encounter a square planar compound, you have more of a tendency to see a pronounced feature in the rising edge. You see that happen for a variety of reasons. I think multiple long-range multiple scattering, where you can think of square planar multiple scattering components coming in and playing a part in that. Certainly FEF-based calculations show that. And also that you can invoke shakedown transitions, which could contribute to that. So I don't exactly know or have an ability to tease out why it's happening, but we know that this type of increase in intensity is typically when you have a more square planar species. And I didn't go into that a lot, but from DFT, you can see that this nickel P is slightly out of the plane in this form, but here it's sitting smack in the plane and it's more square planar.
And redox state of the nickel, because here you have nickel P plus question mark. So
right. So the redox state of that. So that's if you go back to the Endor and Mossbauer, we know that the spin state does not change. So we start from nickel 2+ and we go to nickel 2+. And all of these calculations are done assuming that it is nickel 2+. Now, within that formal oxidation state, you could have charge distribution, which makes this slightly more positive, but I don't think it's helpful to talk about that. I mean, we have trapped, for example, we have trapped this state. We are saying we have trapped this state, and it's in the nickel 2+ state. What was curious for us was that, okay, if we start from here, which is this molecule, which is these distances, and you take away an electron, you oxidize it, you could take that electron out of the nickel, or you take the nickel out, electron out of the iron sulfur cluster. And what we see theoretically is that you're pulling the electron mostly out of the iron sulfur cluster and not from the nickel side.
Okay, so basically, just to summarize that, I have a kind of memory of that system. So you were always detecting something very close to nickel 2+, but the coordination environment changed dramatically, and those details you can delineate.
Yes, exactly. And a lot of that details were needed. DFT was needed to understand those details.
Thank you.
Excellent. You should.
Okay. Okay. So I wanted to go into this. I don't know how much time I will have, but I wanted to give you this example. I was hoping some of the audience would like to hear more things outside biological systems. So the idea here is to combine XAS and DFT to design better electrocatalysts. And we are specifically focusing on homogeneous electrocatalysis. And that's the very promising technology where you can take renewable sources, which are intermediate and weather-dependent energy sources, and convert them to chemical fuels, which can be used whenever we want. And the idea is to improve and develop new electrocatalysts. And the key is that these successful electrocatalysts operate at low overpotentials and high Faraday efficiencies. Again, I rushed through this introduction, but we don't have enough time. So I collaborated with Bob Weymouth at Stanford. They were looking at this cobalt-based hydrogen evolution catalyst. And there was a literature precedent where they started with a cobalt-3 compound, which is bound to a cyclopentadienyl ligand. They do a 2-electron reduction of this. You form a cobalt-1 species, which then binds a proton, forms a cobalt-3 hydride, and eventually does hydrogen evolution, right? So this is a very efficient HER electrocatalyst, but it operates at substantial overpotential. As you can see, you have E1 and E2, they're pretty high. And so what Bob started off doing was to say, okay, can we change this ligands around this cobalt center and make it have a lower overpotential, right? And so what he did was change this phosphine ligand, or diphenylphosphinoethane ligand, to a phenylazopyridine ligand, and said, let's look at this. Does this do HER catalysis? And what they found that the 2-electron reduction happens simultaneously and quite efficiently. You can see it's at -0.17 volts relative to the very low potentials of this system, right? And so it was a strong contender for HER catalysis. But it turns out that despite this promise, no hydrogen evolved. And so they wanted to know why there was no hydrogen evolving. And they knew that protonation was occurring. So they took this and threw a proton in. The protonation was occurring, but they didn't know what was going on. So we started looking at these two systems using X-ray absorption at the cobalt edge. And so I'm going to explain this a little bit. You start again from the cobalt-3 species, you do a 2-electron reduction, you form a cobalt-1 species, and you throw in a proton, and the proton goes and forms a cobalt-3 hydride. This is known, this is the literature precedence. We start with, again, with a cobalt-3 species, do a 2-electron reduction, you throw in a proton, and you don't know what's forming, right? So I'm going to drop the XAS spectra for both of them. You start with cobalt-3, you do 2-electron reduction, and you go right back to cobalt-3, which is what we are saying, this is the cobalt-3 hydride. In this system, you start with cobalt-3, you do a 2-electron reduction, and you stay cobalt-1. So the protonation is happening, but your molecule stays cobalt-1, yeah? And that's interesting. So while we were doing this study, they actually trapped the crystal structure of this molecule. And I'm going to just drop the crystal structure here. It's a little difficult to see this and map it to the molecule that we're looking at. So essentially, instead of the hydrogen binding at the cobalt center, forming a cobalt-3 hydride, the hydrogen actually protonates the nitrogen of the ligand. And because it protonates the nitrogen of the ligand, you really don't see much changes in the Exas. There's some structural changes, small ones, but not big. So what we are saying, you start from this, you expect this to happen, but actually this happens, right? And so the questions here are, why are the reactivities different? One case you form the cobalt-3 hydride, and the other you form a cobalt-1, protonation of the ligand. And at this point, you were saying, can theory help understand this difference and then leverage it to develop an efficient catalyst? Okay, so before we could apply theory, we first wanted to look at all the XAS data and try to replicate the structure that we get from the XAS data with theory. And so we did that. We calculated all 3 molecules, and we made sure that they matched the XAS analysis. And then we did, the other thing we did was we calculated the pre-edge region again. And this is a little difficult to see because these stars are additional features in the DFT calculations. But more or less, the DFT was good enough in simulating the Zanes region. So it could predict both the electronic and geometric structure. Now, if you look at the electronic difference, I'm going to drop all of these together. We then started looking at the charge distribution between the cobalt-1 species of the phosphorus-based ligand and the cobalt-1 species of the nitrogen-based ligand. And you can see there's a big difference in charges. The cobalt-1 is very negatively charged in this, but here the nitrogen is very negatively charged. So this proton, which is happily coming into the system, is actually attracted by this nitrogen. It's obvious when you look at the charge distribution. The other thing that that comes out of the DFT is that if you look at where the charge density is, if you look at the orbital that is going to go and grab this proton, when you look at this molecule again, the phosphine-based ligand, all that density sits on the cobalt. So the cobalt has a negative charge and is primed to grab that proton, but in this other ligand system, which is this, the spin distribution is completely distributed on the whole ligand, and the proton then goes and grabs onto the nitrogen. And we see when we compare these two XAS spectra, they're plotted here. You can see this is reduced, cobalt-1 is reduced, and you do see the shift in energy to lower energy of the rising edge. So this matches the Hertzfeld charge distribution. So, I'm completely out of time, so I'm just going to go and say that, summarize this by saying we looked at the ACS catalysis, and then we looked at the hydrogen evolution by cobalt-based catalyst. What we've done is we have actually gone and designed a first principle electrocatalyst that would work really well, and Bob has tried to synthesize some of them with some success, but I don't have time to talk about that. But essentially in this system, we could specifically say why one system works, why it doesn't, and from there tell Bob that the next ligand that you use should not have the ability to delocalize as a pyridine did. So in the phosphine ligand, no delocalization, cobalt-3 hydride. In here, there's lots of delocalization, no cobalt-3 hydride. Okay, so I think that's the end. I want to stop with this. X-ray spectroscopy, XAS, DFT is magic. Everybody should do this. And I'll skip this and just thank all the people who worked on this. Steve, his postdoc Mehmet, Bob, and Liz, who was the graduate student, and my two postdocs who worked on this in the past. Thank you. And I'll go back to the cobalt-based catalyst and get your questions on that.
That was a terrific talk. Thank you. I think we'll do one last question. So for XAFs, for mostly inorganic systems, whether in catalysis or elsewhere, machine learning methods are making a lot of inroads in giving interpretive ability that humans often don't have by themselves. In bio-XAS, has ML been making much noise?
So it is very interesting that you asked this question. We recently submitted a proposal to ask her to do exactly that. It was a very big team, and I think we won't get funded. The bottom line is I don't think we will get funded because there was some really cogent questions that were brought up, is that we just don't know the protein effects on catalysis, and that's just a very big challenge to take up with ML yet. And so we were told to come back. We had in fact picked up ion sulfur clusters as our starting, because they're so easy from an XAS point of view and other spectroscopies. And they said even then, like you need to define exactly what you will do and limit the scope. So that's what we will do. But yeah, there isn't any precedence yet on that.
All right. But you feel that there's some vistas for it?
Yeah, I think so. I mean, won't it be cool if we could design enzymes which will have, where you could tune the redox potential, right? We say change that amino acid over there and the redox potential changes by this much. That'd be awesome.
Okay, very good.